I'm going to use Star Wars as a way to help explain biochemistry. So I'm hoping you're familiar with the Star Wars saga because it will make it easier for you to understand what I'm saying. Biochemistry starts with cells. A cell is a self-contained unit. It has within it multiple enzymes, proteins. It has DNA inside. It also has this sort of capsule. Everything's inside a capsule. So the Death Star is like that. It's a capsule. It has within it many workers. And those workers each have specific functions. That's the essential nature of this. In the Star Wars saga, the Death Star is evil. We're going to imagine that this is an evil cell. Evil. Which could be a bacterium that causes disease. Certainly not all cells are evil, but it'll help in this case to think of this as a bacteria that we want to kill. Humans are made of cells, and you can see here the scale of a human cell is much bigger than a bacterium. This is a bacterium here. So they're tiny compared to human cells. And that'll be somewhat important as we think about a bacteria being a very simple form of life. But imagine now that's a Death Star. Maybe this is a whole planet, huh? So that gives you some sense of scale. We're going to be interested in this concept, antibiotics. I'm sure all of you have taken antibiotics sometime in your life. It helps you get better. It helps clear the infection. So bacteria can be pathogenic. That's why I'm suggesting think of that as an evil cell. And then you take some medicine to make it so that those bacteria will die. So here, if the Death Star itself is the bacterium, you can see in this case we have a bacterium that's not completed its outer defenses. So the idea of a cell wall is a kind of a protective layer on the outside of bacteria. And if the bacteria's cell wall is incomplete, then they're much easier to kill. So you can see here, penicillin antibiotics, one of the most common types of antibiotics, makes it harder for the bacteria to complete its outer defenses. So here we have some lack of defense. Easy to kill this cell when it's not complete. What will be difficult for you to understand in the beginning is this idea of small molecules. We'll see in the next slide an idea of what does that mean. Okay, so here's a picture of representing a cell. That's the Death Star. I'm making this very simple cartoon showing that the bacterium will create a defensive wall, and it does this using enzymes. So there are enzymes within the cell that will build the wall. An antibiotic, something like a penicillin molecule, is very tiny compared to any of these structures. But what it does is it interferes with the enzyme that is building the cell wall. So an antibiotic, even though it's small, can inhibit or stop the functioning of an enzyme that is essential for the bacteria's survival. So here we go. I'll label this. This is the cell wall. This could be, for example, a penicillin molecule. If the cell wall is incomplete, the bacteria can be destroyed fairly easily. So here's the X-Wing fighter. Luke had a really hard time destroying the Death Star. He had to use the Force. But if you can imagine the Death Star being incomplete, then you could very easily destroy it without needing Jedi mastership. So this is the nature of antibiotics, is to make it so that the Death Star is vulnerable without its cell wall. Exclamation point. Okay, so now we get to the interesting part, because there's this evolution. Humans and bacteria are co-evolving. Humans have developed a weapon against bacteria. Bacteria will evolve a weapon to neutralize the effectiveness of antibiotics. And this is something that occurs naturally. It turns out to be a major problem in modern medicine because bacteria can fight against. Okay, so here we have soldiers that are what they really are in this model is they are soldiers that destroy antibiotic molecules. Exclamation point. So here we have many Stormtroopers in this model. Each Stormtrooper represents an enzyme that will protect against antibiotic molecules. So let's go back to the model here. Here's the Death Star. Stormtroopers are produced. They're cloned. They're cloned within this structure, which is a cell. And their function is to destroy the antibiotics. You remember the antibiotics are these little molecules that are sent to destroy this cell, but the cell produces something that can destroy these antibiotic molecules. An enzyme is a protein. I'm hoping that is meaning to you. Proteins are made of amino acids. And so the cell makes Stormtroopers, makes enzymes out of amino acids so that the cell, can survive even if antibiotics are sent to kill it. Alright, so penicillin is a small molecule. We talked about that before. It's actually quite fragile. If you wanted to destroy it in a test tube, you could easily do that in 15 minutes. Throw some strong base in there, warm it up, and penicillin will be gone. The cell, instead of using chemical means, uses enzymes. So the Stormtrooper here represents a beta lactamase enzyme. It seeks out penicillin molecules specifically. It can destroy them easily. So this one Stormtrooper might say, there's a penicillin molecule, zap, got it, dead. There's another one, zap, got it, dead. These Stormtroopers have good aim. They are able to find their target specifically and destroy it rapidly. Here's a cartoon that may help you understand the sort of the scale. So, scale of the cell. Here's the cell. This is the evil bacterium. It's our Death Star. So it's big. Big. That's no moon, but it's big. This tiny molecule, molecular weight 400, that's the antibiotic molecule, like a penicillin molecule. And the beta lactamase enzyme, this enzyme here, made by the cell, is designed to intercept the penicillin and destroy the penicillin before it can harm the cell. So it's kind of an interesting story. It would make a pretty good movie. The penicillin is defenseless. If the cell didn't have the enzyme, then the cell is itself defenseless against penicillin. Kind of interesting. So this tiny molecule can kill the cell unless the cell has this defense. And so that in our model is the Stormtrooper. So the cell makes Stormtroopers. The Stormtroopers protect the cell against the penicillin. I'm hoping you can kind of see this enzyme is made to eat up that particular penicillin. Enzymes are often represented by the Pac-Man icon. This is the actual structure of penicillin. This part right here is the unstable part. It has this four-membered ring. This is an electrophile right here. The way it works to kill a cell is this reactive carbon here. This is a carbon. Will actually become bonded to the enzyme that builds the cell wall. So the enzyme that builds the cell's wall will become bound to this carbon, and since that occurs at the active site of that enzyme, the enzyme is then useless. So the wall doesn't get built, and then it's open to attack from the X-Wing fighters. It actually doesn't even need an attack. It'll just fall apart without a cell wall. That bacterial cell is toast. The lactamase enzyme will cleave this bond right here. The lactamase enzyme will cleave this bond, and therefore the four-membered ring that was essential for its function is destroyed, and it becomes useless in the war against bacteria. So the enzyme destroys penicillin. Okay, now this is the way that scientists often represent a protein. There'll be an amino acid here, and another amino acid here, and another one here, and another one here. And so this is actually a continuous chain. If you trace it, it's kind of like a maze. But it'll be a continuous chain. Happens to be, for this particular enzyme, 263 amino acids. That will then fold up in particular structures. This is called an alpha helix, which is a common secondary structure in proteins. The blue arrows are called a beta sheet, another common secondary structure in proteins. This entire structure, 263 amino acids, is designed to chomp this tiny molecule here. This is penicillin, and this big structure is designed to chomp that penicillin molecule, rendering it useless. So maybe you can understand this nature of representing the protein, the enzyme, as a Pac-Man, and its target as a little molecule here which can be destroyed. It'll cost the cell some energy and some molecules to make this. So from this economic standpoint, the cell has to get some benefit out of the investment. It's investing 263 amino acids, and it's also investing a whole bunch of ATP to put them together. But in this case, it's a good investment because it confers survival upon the bacterium. This is showing you gene sequence. There are 4 bases in DNA: G, C, T, and A. Guanine, cytosine, thymine, adenine. And when you want to build a protein, you need what's called a gene. This gene starts right here. ATG is always the first 3 letters of a protein, well, of a gene that's being translated into protein. That stands for methionine. That's called the start codon. Start here. This is a lysine, serine, leucine, serine, alanine, threonine, leucine, isoleucine, serine, alanine. So you can see that every 3 DNA bases codes for 1 amino acid. 263 amino acids makes the entire enzyme. So it ends right here. There's what's called a stop codon. That's the end. So that, in order to make this enzyme, the bacteria must have this gene sequence. Not every bacterium has this gene, which is good, because if every bacterium had the gene, then every bacterium would be able to destroy penicillin and your antibiotics would be useless. So it is possible to transfer genes between organisms. Bacteria do this naturally. Scientists can do it unnaturally. And so the bacteria we'll be using have been given this gene, have been given the gene that destroys penicillin. And that allows us to study the enzyme that they make. Okay, that's the end of the show. Let's go back and see whether everything makes sense. This, I think, is the most important one. This cell has a gene, some DNA that allows it to make this enzyme, the beta lactamase, that will protect it, the cell, from penicillin, which would otherwise be fatal to the bacterium. In this project, we're going to try to isolate this enzyme. It's not inside the cell. It has to come out to be able to intercept. It's like a TIE fighter sent out from the Death Star. It's a TIE fighter sent out. It's going to intercept the penicillin out here in space. That'll make it easier for us to isolate the enzyme, because we don't have to get it out of the cell itself. We're going to get this. It's a protein. It has a molecular weight of about 30,000. And we'll be able to measure its activity. We will feed it penicillin. And you will see spectroscopically in the UV the destruction of penicillin by this enzyme as it goes chomp, 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 chomp. We'll be able to follow that by measuring changes in the UV spectroscopy. This project should work well. The problems that students often have is they don't treat things with proper respect. Penicillin, it's unstable, but if you leave it at room temperature for a day, it'll still be there. Enzymes are fragile and they must be refrigerated. And so one of the challenges we have here is students tend to leave things out of the refrigerator and they come back tomorrow and their enzyme is dead. The other challenge we have is we're going to be interrupted by Christmas break, winter break, and you'll be gone for 2 weeks, and you hope that your enzyme survives those 2 weeks. So you'll learn a lot of new things, gain some new skills, and I hope you'll be able to appreciate the wonder of this, that life finds a way.